Hello, my name is Teresa Snyder-Leiby. Welcome to today's GeneMarker HID Human Identity Software Short Tandem Repeat Mixture Analysis webinar. GeneMarker HID software is an expert system used by forensic laboratories for human identification. During this webinar, we will use STR mixture data from the National Institute of Standards and Technology website to walk through all of the mixture analysis options in GeneMarker HID. Please review the introductory GeneMarker HID webinar for details on user management, importing data, setting analysis parameters, reviewing results, editing results, audit trail, and reporting options. As a reminder from the introductory webinar, GeneMarker HID has been validated by forensic labs worldwide and is concordant with their previously used genotyping software, such as GeneMapper, GeneMapper IDX, and GenoTyper software. GeneMarker HID is compatible with all CE instruments and human identity chemistries for rapid, accurate size and allele calls. The user-friendly linked navigation decreases analyst strain and saves time. GeneMarker HID has automated control and allelic ladder concordance checks and the ability to customize parameters for each locus, including RFU for the analytical range, stochastic range, and stutter filters. The user management provides control of access rights and an audit trail. GeneMarker HID has several reporting options, including print reports, tables, and CODIS formats. During today's webinar, we will briefly review the process of importing data and optimizing a panel for mixture analysis. After setting the mixture parameters in the panel editor, we will apply the panel using the three-step run wizard, detect mixture files, and discuss quantification and reporting options. During the second half of the webinar, we will review the embedded mixture analysis application, which automatically will detect for all mixture samples the number of contributors to each mixture sample and calculate the PE, CPE, PI, CPI, and RMNE. For two contributor samples, GeneMarker HID reports all single source files that are potential mixture contributors, automates repetitive calculations for all possible allele combinations, and identifies major and minor contributor genotypes based on mixture ratios. The program calculates likelihood ratios for each locus and the combined likelihood ratio. We are able to submit profiles to the searchable database and search the database with a profile, whether it's a single source profile from the current program or a genotype that was deduced from a mixture. As a brief review from the introductory GeneMarker HID webinar, GeneMarker HID accepts files from all major capillary electrophoresis instruments, including FSA, HID, RSD, SCF, and SG1 formats. For mixture analysis, it is generally necessary to make the parameters more sensitive to call minor contributor peaks. An authorized user will have access rights to make the needed edits to the panel and mixture analysis parameters. As we saw during the introductory webinar, each marker uses customized parameters. Activate the settings box with a right mouse click on the marker name above the electropherogram. The minimum homozygote and heterozygote intensity is the minimum RFU or height that will be called within a marker. These are used to set the analytical range. The inconclusive range is used for quality flagging of peaks that are above the analytical range of the instrument but may be subject to allele dropout, or the stochastic range. Peaks that are above the minimum intensity and below the inconclusive level will be flagged yellow during the analysis, and the quality reason will be IHO or IHE. Any heterozygous peaks that are below the maximum heterozygous imbalance set here for the marker will be flagged yellow with IMB for the quality reason. An essential parameter for mixture analysis is the minimum heterozygous imbalance. Peaks below the minimum heterozygous imbalance are not called. In other words, the minimum heterozygote imbalance setting acts as a filter rather than a quality flagging parameter. A typical setting for single-source samples is a 20% filter. This will miss minor contributor peaks. It is essential to set the minimum heterozygous imbalance to a level that will detect minor contributors. We have selected 10% for this analysis. Any peak that is 10% or more of the tallest peak in the marker will be called. To call all peaks within the marker and have the analyst review each peak individually, set this filter to 0. The stutter filter should be set to values determined by your lab for stutter at each marker. Panels with parameters customized for mixture analysis should be saved as new panels. Using the path File, Save as New Panel, input unique name. In this example, I used MX to indicate mixture parameters and 3100 to indicate that it has been saved with the signal information or calibrated to an AB 3100 genetic analyzer. There are several display and reporting options. Before starting, select the settings that are preferred in your lab operating procedures for labeling the peak ratio by height, RFU, or by peak area. Leave the panel editor and activate the Run Wizard using the Project dropdown or the green arrow icon in the main analysis window. In the first screen of the Run Wizard, select the panel template with the appropriate parameters for your mixture analysis. Please see the introductory GeneMarker HID webinar for details about creating and saving Run Wizard templates. Confirm the settings in the second screen of the Run Wizard and then click Next. In the final Run Wizard screen, select Mixture Evaluation and the peak percentage. In this example, files with peak ratios where the minor peaks are at least 10% as tall as the highest peak in a locus will be detected as mixtures. And set the minimum mixture per marker number. In this example, any files with 3 or more peaks per locus will be detected as a mixture. Then select OK to run the analysis. Analysis is rapid, generally processing 96 samples in approximately 20 seconds. In the file name tree at the upper left, we see that the last 2 files are consistent with the parameters for mixtures set in the third screen of the Run Wizard and are indicated with MX: and olive green font. The allelic ladder has a few virtual alleles with higher than expected RFU, as indicated by green allele labels and yellow vertical bars. The electropherogram and allele report table for the MX Case 2 evidence file has red flagged alleles at the minor peaks. As seen in the peak table, the quality reason is PL, greater number of peaks than expected for the ploidy setting of the panel template. If you prefer not to have these samples flagged red, simply change the ploidy of the panel template to unlimited. This will result in just the imbalance rule being fired for yellow flagging, as is seen here for the Y allele of the amelogenin marker.